Welcome to another tutorial of a video series on genome-wide analysis. In this video, I will show you how we can perform evolutionary analysis of a protein family, and we will be finding which proteins are orthologs and which proteins are paralogs, in addition to finding speciation and duplication events in the genome. So let's begin. So as you know, in the previous video, we have already done with the figure 1 and figure 2. And as you know, in the figure 1 and for figure 2, which were gene structure analysis and domain analysis, we have already downloaded proteins from Arabidopsis genome and from the rice genome. So we need to make sure whenever we need to perform evolutionary analysis, we should have must two sets of proteins. Like here we have downloaded the bromodomain gene containing family of proteins from Arabidopsis genome, and this one we have downloaded from rice genome. So if I can show you, here you can see in this file we have actually the original ID. So we also need to use this file in which we have the original gene IDs. Here you can see this file we will be using, and we will be using another file that is from the Arabidopsis genome. So here in the Arabidopsis genome, this one you can see we have also the original gene IDs which we have downloaded in the previous video. So if you don't know how to download, you can go back and watch the previous videos first. So we will be using these two files for our evolutionary analysis. So let me make another folder which would be the evolutionary analysis. So we will be finding orthologs and paralogs. So I can rename this folder and I can copy this one, all proteins from Arabidopsis genome, and similarly we can copy this. So I can simply copy this, I can take it to this folder and I can simply paste it here. So we can go to Google and find one online tool which is recently published, which is Orthovein 3. Although the previous version was Orthovein 2, but Orthovein 3 is a bit more efficient in finding the orthologs and paralogs. So I recommend you can go with this Orthovein 3. This website is totally free for research purpose, so we can simply open it. So here you can see by using this website, we will be able to find how many orthologs of this family in Arabidopsis and rice genome. So first of all, we can go to this start tab. And if you can see in the start tab, here are the different options like metazoa, plants, fungi, vertebrates, protists, and bacteria. So we are actually choosing plants. But if you want to perform analysis at the whole plant genome level, then you can choose this plant. From here, any species you can select. But as you know, we are only choosing this bromodomain family, which we are actually reproducing the result of this already published research article. So we will be using this as a reference. So as here in this figure, you can see they have actually drawn different orthologous groups. And here, even if you can see in this figure, when they have drawn the comparison of gene structure, you can see they have orthologous group 1. If you can see, here are 1, 2, 3, 4. There are the 4 genes from Arabidopsis and 1 gene from rice genome. So these 5 genes are actually in the orthologous group 1, and similarly 1, 2, 3, and 4 are also in the orthologous group, and 1 from rice genome. So we will be reproducing this result, and I will show you that how we can also find this orthologous groups, paralogous groups, and singleton categories. So we can go, because as you know, we are not comparing the whole level of plant genome, so we will be only uploading our proteins. So if you can see, it is only accepting the FASTA file. So I can go back to this folder and which we have just created. So what we need to just change this text format into FASTA format. So we can simply go to rename option, and we can change this text to FA. It would be the FASTA, and we can also change this text version to FASTA. So that's it. We have changed our files from text format to FASTA format. So now we can download them. So we can simply click on here, and we can click on this orthologs and paralogs. And first we can choose Arabidopsis, and we can simply, you can see here the list is chosen. So we can simply add it to analysis list. So you can see it has been added to our analysis list. So now we can choose the rice bromodomain all proteins. So we can also add this to our analysis. So here we need to set our analysis configuration. So you can see for orthologous analysis, we are using OrthoMCL algorithm, and we can also use OrthoFinder algorithm. So I will recommend you to go with the default and e value, that is 1 e power -2. So that is 0.01. So I also recommend you to do not change this. So we can also go with the phylogenetic analysis, although here we will be mainly focusing on the orthologous analysis, but you can also check on this phylogenetic analysis, collinearity analysis, expansion and contraction analysis. But if you will choose this collinearity analysis, so for this maybe you need to upload GF file. So I will uncheck this because we are mainly concerned with orthologous and paralogs finding. So I can simply task this name. So we are creating this for video, so I can simply write video for YouTube, and here you can write your email, and you can simply click on this start analysis. So you can see it is showing that it is uploading files. Please do not reload page. So now it is computing the orthologs by OrthoMCL algorithm. So all these steps will be finished soon, as long as we are only uploading our small files. But if you will be performing orthologous analysis or evolutionary analysis at the whole plant genome level, then it may take up to even days. So now it is performing orthologous overlap annotation and cluster go enrichment. Here you can see the up to 6 are already done, so we can wait a bit more. So now it is calculating the evolutionary relationship between the species, and that's all. Here you can see now we have done with our orthologous analysis. And in the orthologous analysis, actually there are the two methods. One is the occurrence based, and one is this one. So you can choose with anyone, both the results are same. So if we can see here, you can see there are actually the 13 cluster of, here you can see versus in our first file was from Arabidopsis all proteins intersection rice all proteins. There were 13 clusters. So actually if you can go into this research article, so you can see they have also 13 orthologous group: 1 2 3 4 5 6 7 8 9 10 11 12 and 13. So here you can see we have also 13 orthologous groups. Here you can see one was unique to Arabidopsis genome, and this one was unique to rice genome. So if we take the chart view or the table view, so we can see there were actually in the Arabidopsis there were actually 27 proteins which were divided into 14 clusters and 2 singleton categories. And in the rice genome you can see there were actually the 22 proteins which were divided into 14 clusters and 4 belongs to the singleton category. So we can go back to this one. So here you can see we have actually 14 clusters. So among them, common between the rice and Arabidopsis are 13. So here you can see here we have 13 group, one belongs to Arabidopsis genome and one belongs to rice genome. Here you can see it is also showing the same results. Number of elements were specific to 13 and 13 belongs to two species, and there were two sets which were only belonging to one and one species. So how we can know that which genes are orthologs and which genes are paralog? So we can simply go to this download button, and here you can see here we can download the results. Here are the co-orthologs, here are the paralogs, and here are the orthogroups, and here are the orthologs. So you don't need to worry about what are the orthologs and what are the paralogs. When I will show you the results, you will automatically know that what are orthologs and what are paralogs. So first of all, we can download those groups. So we can simply download first this one. So here you can see our file is downloaded. So this is G file, and here are actually the orthogroups. So you can see we have downloaded the orthogroups. So we can count them: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and 15. So here are totally 15 groups. Among them, there are 2 paralogs and 13 orthologs. So here I can tell you that both orthologs and paralogs belongs to the homologous category. So they actually share the common ancestor, but actually the orthologs are those groups in which the speciation has occurred, and the paralogs are those groups are those genes in which the duplication event has occurred. So if we take our first, so you can see the first gene is from the Arabidopsis, second gene is from Arabidopsis, third gene is from the Arabidopsis, and fourth gene is from Arabidopsis, and the fifth gene is from rice. So it means this is the first group. Actually, this is the first orthologous group. So if I can show you in this one, we can go back here and I can show you. This was actually our, I can zoom in. So here you can see one. Actually, you can see the one belongs to rice group. Here you can see 1, 2, 3, and 4 belong to Arabidopsis genome. So as long as there are two species, so it means actually the speciation has occurred, so they belongs to orthologous group. So let me show you with the IDs. So if I can show you, in the first group are actually 5, and in the second group you can see in the here are the, again in the second group you can see in the second group there is one gene from the rice genome, and 1, 2, 3, and 4 belongs to the Arabidopsis genome. So if I can take you here in this example, so actually the first group they have divided. Let me zoom in. So actually there were the 4 genes. You can see these 4 genes. We have now actually given the name. This was the ATBRD1, actually because it belongs to the orthologous group 1, 2, 3, and 4. So we have given the name A, B, C, D. Actually because they are belonging to the one orthologous group, so we have given the name 1, 1, 1, and we have just to make it different, we have given name A, B, C, and D. And if you can see, there is only one gene which is OSBRD1. So you can see in this way we can give them name according to their orthologous group. So if I can take you next, here you can see here is another orthologous group. So we can take this one. Here you can see this is also from the Arabidopsis. This is also from Arabidopsis. This is also from Arabidopsis. And here you can see one is from the rice genome. So it means 3 are from Arabidopsis genome and 1 is from rice genome. So it means you can see because they are different species, so speciation has occurred. So it means they are orthologous group. So here in this you can see this is from the rice genome, this is from rice genome, and this is from rice genome. So you can see all three belong to rice. So you can see rice is only one organism, so it is not sharing. So it means actually the duplication event has occurred, so they will come under the heading of paralogs. So I can show you in this figure that they are actually paralogs, 3 genes. Yeah, here you can see we have 3 genes. This one, rice, so this is actually belonging to the paralogs groups. So we can go to the next. Here you can see one is from the rice, one is from rice, and one is from Arabidopsis. So this would be the orthologous group because 2 are from rice and 1 is from Arabidopsis. So they will be the, because this was the actually the paralogous group, and this would be orthologous 1, 2, and this would be the 4, and similarly orthologous. So in this way you can have an idea what are orthologous groups and what are paralogs group. So for an example, we can see here, you can see in this one we have actually the one is from the rice and one is from Arabidopsis. So what it would be, whether it would be the ortholog or the paralog, because it is sharing the 2 different species, so it means the speciation has occurred, so they are the orthologs. So but if I can give you another example, here you can see this one. This one is Arabidopsis, this one is from Arabidopsis, and this one is also from the Arabidopsis. So they are sharing only one species, so it means the duplication has occurred, so it belongs to the paralogs group. So in this you can see the 2 Arabidopsis, they are belonging to the paralogs group. So here you can see they have actually drawn these structures. So how we can also draw on this type of the structure. So we can go to this one, and we can actually go to their clusters. So we can simply click on this 13, so it will open all those clusters. So here you can see we have list of those cluster. So in the first cluster we can simply open it in the new tab. You can see in the first cluster there are actually the 5 genes, so you can simply take its screenshot. You can see 1 gene is from rice. You can even change its structure, and there 4 genes from the Arabidopsis. And similarly, if we see the cluster 2, you can see there are actually 1 gene from the Arabidopsis, and 1 gene is from the rice and 4 genes from the Arabidopsis. And similarly, if we can see cluster 3, so they are actually 3 genes from the Arabidopsis and 1 gene from the rice. So in this way, now you would have a clear idea that how you can give name to this structure on the basis of this orthologous group. So here you can see here we have orthologous group, and this orthologous group 4 from Arabidopsis, 1 from rice. And similarly in the orthologous group 2, 4 from Arabidopsis, 1 from rice. And similarly in the orthologous group 3, 3 from the Arabidopsis and 2 from rice. And similarly 1 from Arabidopsis and 2 from rice. In this way you can just make their group. And now I have already told you about how to find orthologous group and paralog group. So how you can find singleton categories. So you can go back here, you can go to this download, and here are the single copy clusters. So we can simply click on this download. So here are the singleton. There are these cluster which can have the different genes. You can see these genes do not belong to paralogs or orthologs, so they belong to singleton categories. So if you also want to download the orthologs one by one, so you can also download. You can see here we have the which genes are orthologs. So as we know that for orthologs, the speciation should occur. So one is from the rice, you can see one is from the Arabidopsis. So here are the different pairs of genes which are orthologs to each other. So hopefully in this way you can find orthologs, you can download them, and you can now divide your whole set of bromodomain family into their orthologs groups and the paralogs groups, and similarly to their singleton categories. So if you can see in the singleton categories, from the Arabidopsis were only one gene, and from the rice there were only 3 genes. So if you also want to have a look on their gene ontology, that this 13 cluster are actually coding for what type of biological processes. So you can see in the cluster 5 there were actually 5 proteins which were essential for meiotic recombinations. So we can also find a list of biological functions, molecular functions, cellular components, and similarly the Go enrichment can be used for this cluster analysis. But this part maybe is not required for genome-wide analysis, but it would be even more useful that if you can add this type of information, having biological processes, molecular function, and cellular component into your research article. So there are the next tabs, which are the phylogenetic analysis, and here is expansion and contraction analysis. So we will be focusing on these two type of analysis later on in the separate videos. So if you have any question related to finding orthologous groups or paralogous groups, please let me know in the comment section. Thank you for watching. Bye bye.